Yeah, definitely. I mean, 100 patient clinical trial, right, for their pivotal trial, each batch being, at least 1 million plus, right, for each patient. Um,
Wow.
just do the math on that.
It's more zeros than I'd like to consider.
Yeah.
Absolutely.
I mean,
Wow.
what's
It
in
is it's good news,
I had
right? It's good news. We don't necessarily know exactly all the details, but I think it's good news.
yeah.
I took it as great news. I mean, I hadn't seen that kind of data, on the manufacturing cost. On somatics autologous, everybody talks about the high cost. Manufacturing is not these days such a huge driver of the price, as big a driver of the price as it used to be. I don't think. I think the manufacturing cost is coming down. We're having some arguments about, I think, about the value pricing.
Mm.
But if the manufacturing cost, Jonathan, you're saying is $1 million, that's logarithmically different.
Yeah, yeah, absolutely. If we're talking about a autologous iPSC approach, right?
Hmm.
Yeah.
Yeah. So
Que- Questions whether that goes in vivo as well.
absolutely. I think that's definitely a very, very salient point, and it'll be an interesting one to see as we move into ASH. So, welcome everybody. It's wonderful to have so many of you join us, and welcome back to Advanced Therapies Think Tank, or AT3 as we like to call it. We're really, really glad that you're here, and we're glad that you've been able to join us, and hopefully you are live. So if you are new to AT3, we are a solution-focused platform really for experts across the advanced therapies ecosystem to tackle big, big challenges head on. The way we do this is we skip the primers and we dive straight into the really, really big questions because we know you're already working in this field and we know you care. Each episode is built around a single pressing topic. We bring in expert voices from the industry as investors, payers, regulatory, and patient-facing roles, and we aim to challenge assumptions that are out there and create real, actionable ideas. Our faculty helps us to shape these discussions throughout the season, and we'll close each session with key takeaways. But before we get started into all of the good discussions, none of this would be possible without our seasons and session sponsors. So I'd like to take a moment to extend a really huge thank you to our season sponsor, BCA Advanced Therapies Network, and also to our session sponsor for this session, Scale Ready. And Scale Ready is a leader in practical, scalable solutions for ex vivo cell therapies. Scale Ready works every day to help autologous cell therapies become more viable and more accessible, and really, really topical for today. Their focus is on simplifying processes through technologies like G-Rex and reflects a deep commitment to the strengthening of the foundations in the cell and gene therapy ecosystem. It's this perspective and their drive to expand hope for more patients that make Scale Ready such a valuable supporter of discussions shaping the future of advanced therapies. Right, so let's get into things. Joining me today are two faculty members. First of all, Carl June and Miguel Forte. And also, I'd like to introduce our invited guests, Cécile Bauche, JP Latere, and Jonathan Ye. Now, before we get stuck into the discussions for this session, we have also partnered with Beacon Intelligence to provide exclusive data and insights to support our discussions. The team at Beacon has very kindly shared data and analysis around this topic, and so we'll start with a really quick review of some of the information collated. We'll only go through a couple of slides, but we will share the entire data set in the chat for you to view at your leisure. So, Georgie, if we could go to the first slide, which worked perfectly when we were testing it. So,
And there we
talking
go.
through this, there we go. It's come up wonderful, Georgie. Thank you very much. If we could maybe go back one, that's it. So, if we look at some of this really interesting data comparing autologous and in vivo therapies, as you can see, as expected, coronavirus infection is really the only massive indication that dwarfs autologous development. But after this, autologous development really is much, much larger than in vivo. Not really surprising. after this coronavirus, the really next big areas, as we might expect, are cancers, with a focus on hematological malignancies, which lead as a narrow margin over solid tumors. Then at the top right, you can see that pie chart. We can also see that in vivo approaches are really starting to gather pace over autologous. And so more on this later. If we move into slide 2, the second slide we'll show, there are no really big surprises here. This one, when it comes up, will show the delivery systems. And as you can see, non-viral delivery systems led by LNPs are really driving the in vivo development forward, for many of the reasons that we'll discuss today and in future sessions. So that's just a little bit of an introduction into some of the data that Beacon has collated, and that should be available for you in the chat. So if we come back to the main session, the session we're discussing today in AT3 really pushes the boundary and will challenge some strongly held beliefs about where our industry came from and where it's heading. Pioneers like Carl June and Luigi Naldini, both AT3 faculty members I may add, have ushered in a second coming of cell and gene therapies with autologous approaches using both cell-based and gene therapies. And these were truly industry and life-changing, and for the first time allowed us to aim for the C word, that's cure. But soon these incredible feats of medicine and engineering were taken over by discussions on price, which, as we know, still drives a lot of discussion. Go back to our second episode of AT3 for some more discussions around pricing. And this then potentially limits patient access. So as the promise of truly personalized medicine dimmed under the harsh light of reality, we began to talk about in vivo cell therapies and gene therapies as the true holy grail, a way to still effect a cure, but at a lower, more manageable set of costs, theoretically, hopefully. As with so many of our big plans, reality and biology often have their own ideas. And so the path to in vivo therapies has been somewhat slower and more complex than the cover of Time magazine would initially have us believe. It's become very clear now that not every cell or gene therapy will be amenable to an in vivo approach. Not really surprising given the history of cell and gene therapies. So this is the nub of our discussion today. What is the future of autologous therapies, and where do in vivo therapies fit in? Or is it maybe the other way around? So through the session, please use the chat and the reactions functions to engage with the panel and with each other. We really want this to be a collaborative and interactive session, and we want to hear what you think. We also have time allocated for audience Q&A. So all of those questions, when they come up for the panel, please submit them using the Q&A widget at the bottom of your screen as we go. And you can also comment and up and down vote these questions as you please. And then Lee will pose them to us at the end. So let's get into it. The first one that we start looking at is autologous therapies are marketed as personalized, but they inherit each patient's unique cell dysfunction, prior treatment damage, and immunological baggage. Meanwhile, allogeneic and in vivo approaches can use optimized, quality-controlled starting materials as precisely targeted interventions. So, are we romanticizing the personalization when standardization might actually deliver superior and more consistent outcomes? Carl, we'll start with you. As the father of CAR T, is autologous ex vivo therapy's biggest strength actually its fatal flaw?
Thanks, Sven. I'm going to be a little controversial. I think we have to remember that we evolved a thymus, and the thymus selects your T cells so that they can survive for a lifetime in you. And at the same time, you reject other cells. We found that it's very hard to fool the human immune system. And there's lots of genetic approaches now with allo cells to do that, with knockouts of class 1 molecules, T cell receptors. But to date, if you have an indication where there's persistence is important, nothing has, the benchmark has been what autologous cells do. And then there's what you mentioned, Sven, the safety issue, depending on, what the patient brings into it. And this is an issue. If the patient's heavily pretreated, they may have very damaged cells that have a different safety profile than a healthy donor. And I'll stop there.
That's brilliant, Carl. Thank you very much. And I think really salient, bringing back to the biology, we so often get so caught up on the engineering, but we forget about the natural biology. So Miguel, I'm going to come to you. And given the inherent manufacturing complexity, as well as COGS and vein-to-vein logistical challenges of autologous therapies, at what point does the total addressable market for these products effectively plateau?
Thanks, Sven. And thanks, Carl, for actually setting the stage. Because what we're saying is there is a biological need opportunity, and we need to preserve that through the autologous approach. Now, you're right, the autologous approach represents a series of complex difficulties, the supply, the clinical aspects, leading to a market that is being growing slower than we would like. So to answer your question, I think the plateau is because of the technology, not because of the need. And we're seeing that slow growth, not plateauing, but definitely below where it should be. How it will evolve depends on how we address those challenges and improve the manufacturing, and more to discuss later, I think, but also how is the penetration of in vivo competing to the autologous. So in the end, if we manage the autologous, if the competition comes in, there'll be a balance between the two, and we will see a plateau, not driven by the technology, by driven the fact that we've treated all the patients that need to be treated.
Fascinating insight. Thank you very much, Miguel. So JP, let's move to you. Theoretically, the in vivo approach represents the ultimate form of standardization by bypassing ex vivo manufacturing and logistics complexity entirely. The success of this model really hinges on highly efficient, specific in vivo targeting and minimal off-target toxicity. So what do you feel is the single greatest non-biological hurdle, example, regulatory acceptance or delivery vehicles, that we are going to face in proving that the standardized systematic intervention can not only match, but potentially surpass the precision and safety profile currently achieved by the personalized autologous therapies?
Yeah. Thank you for the question, Sven. I think the first thing is that the in vivo is super exciting on different aspects. So obviously, I'm a bit biased. I've been working on the in vivo for the last few years. I think really the way we see in vivo focusing on CAR-T, it is that the in vivo allows you to make an autologous product inside of the patient body. So you're really basically reconciling the two words. So from a biology perspective, it's true that I think there are a number of challenges that in vivo needs to solve, and we're working on that. From an isobiotic perspective, when we look at what we did, I think in vivo shows now you can be very specific, you can really transduce at high level cells, etc. So I will say that on the biology side, personally, I feel extremely comfortable because there are so many things that we can do. I think we're just scratching the surface of the in vivo work, but we can discuss that a little bit later. Now, if you're looking at non-biology, I think what I'm seeing as the biggest challenge is probably a combination of the regulatory and the economic framework. Why am I saying that? I think, and it's interesting, initially when I was trying to pitch the idea of the in vivo, talking to investors, I was presenting that as a gene therapy because I didn't want to feel like it was a CAR-T, because at that time people were like, no, CAR-T, we put enough money into CAR-T. So I said, oh, that's a gene therapy. But technically it's not really a gene therapy because with the in vivo CAR-T, we're not changing the gene. We're not doing that. We're using a lentiviral vector to deliver a payload, but we're not changing the gene. But I was positioning that as a gene therapy. And then when there were a couple of news around gene therapy, patient death, unfortunate patient death, and so on, I was like, no, we're more like a CAR-T. So the in vivo position itself in the middle. So from a regulatory perspective, I think we need to work with regulators to make sure that we understand how to position that. Because now, for example, in our case, or in the case of LNPs, you're not delivering a cell. You are delivering a viral vector or a non-viral system. So you have to be able to release that to address the potency and etc. And at the same time, I'm saying it's an economic challenge because the way you manufacture that, those two elements are associated. So you have to be able to really to seize the promise of in vivo. You have to be able to manufacture that at a scale which makes sense from an economic perspective. We'll certainly come back to that, but I think that combination, regulatory and the economic framework, that's where we need to be able to address some of the questions over the next few years.
Really interesting and really great to have that experience. And I think this also speaks to the way we classify these therapies. I think possibly the science is starting to outstrip the classification that we currently use, cell versus gene. So you spoke there about how you positioned an in vivo approach to investors. And so let's go to Jonathan. As an investor, in your portfolio allocation, how do you think about weighing the de-risking profile of an autologous platform, which is established, albeit challenging from a clinical, logistical, and reimbursement precedent, against a potentially higher yet more speculative return on investment and broader accessibility theoretically of an in vivo approach?
Yeah, I think these are, fundamentally two different approaches, two different questions that we're looking at when we assess these opportunities. You made a comment when you were talking to JP about in vivo being the ultimate standardized therapy, but I guess my view of it is that it's actually standardized raw material that you're putting into the patient. Whereas from an in ex vivo context, the starting material is variable, but the drug product, there's a level of standardization that's coming out and then going into the patient. Because there's that standardization there, I think autologous remains, a great reductionist biology tool in a sense, where you're able to look at the CAR construct or ascertain whether a target will be appropriate in a particular patient context, and then be able to hone in on those questions. And I think that's where autologous still remains valuable. Whereas, with in vivo approaches, you've now stacked risk in terms of delivery vector as well as payload delivery as well. And now if you're going to stack on top of that, target validation or binder validation, it becomes a bit insurmountable. So, when looking at these different opportunities, I think you're looking at it from the perspective of, are you trying to go after a new patient population and validate a target? Or are you looking at the platform itself, being able to deliver into new cell types or trying to penetrate into new patient populations using that in vivo approach?
Fantastic. Really, really interesting. So big message so far, autologous is not dead. So let's not forget, we have to treat various different patients in different ways. Let's not throw the baby out with the bathwater. Cécile, coming to you. Very exciting. Autologous therapies really rely on attempting to control the quality of the starting material, as Jonathan was talking about, while standardized in vivo therapies rely on controlling the efficiency and the tropism of the delivery modality. From a core biology and engineering standpoint, which of these two control challenges, ensuring minimum acceptable cellular fitness for ex vivo or achieving a consistently precise, predictable tissue targeting for in vivo, which do you think represents the higher technical hurdle to achieving true superior and widely accessible outcomes?
Thank you, Sven, for this more technical question. It was mentioned earlier that for ex vivo manufacturing, you start with something from a patient and you start with cells exhausted, pre-treated, and it's difficult to predict how they will behave during the manufacturing. And I think if you start manufacturing, it's getting complicated to rescue it if something goes wrong. You have means, you have things, but it's getting difficult. With the in vivo, you move the challenge to the delivery, because as you mentioned, you want to achieve a consistent and a selective targeting inside the patient. I think it's technically demanding, but it's solving by engineering finding some solution based on engineering the vector. So as of today, if I have to compare both, I would say the first one for ex vivo is more challenging, because for in vivo, you have tools, at least on the paper, you have tools to targeting agents, cell-specific promoters, suicide genes if you want. So you can at least start with having ideas and validate them ex vivo. And I think I would add a third control challenge that was not on your question, that will make the question difficult to answer, is immunogenicity. It's something that you cannot predict with in vivo, depending on the nature of your technology and of the patient history. So starting from your question, I will say I think the hurdles are more in the ex vivo part, but if you add immunogenicity, maybe it's becoming about more complicating. And I think you can solve that. You also mentioned something like the dosing, and I think you will have a good in vivo product as soon as you will know the number of T cells that you want to modify in vivo. Because right now people are talking on VP, micrograms, TU, and you don't really know how many cells you will have. And if you start to think about how many cells I'm modifying, you will have something more consequent, and the balance will go through in vivo.
Lovely. Really, really fascinating. Thank you so much, Cécile. So here you have it. So we're looking at that personalization, and I think really this is a case of it's not one or the other, as we so often like to go after in medicine, but there are variations, and so we need to think about the right therapy for the right patient. So let's move more into some of the costs. So we know autologous ex vivo therapies like CAR T have really achieved remarkable clinical success, but very often at costs that exceed $500,000, sometimes even $1 million per patient. Given the rapid advances of in vivo gene editing, base editors, prime editors, epigenetic editing, and a variety of others, and all of the off-the-shelf allogeneic approaches, are we actually starting to invest billions in manufacturing infrastructure that will become obsolete within a decade? Should we rather be directing that capital and talent now to the in vivo, or is that a little bit premature? So Carl, we'll start with you again. As the originator of the CAR T technology, you've seen its maximum potential realized in the clinic, and we've seen some of your patients speaking around the world, absolutely incredible results. But beyond the logistical and manufacturing challenges, do you think that autologous T cells itself, a product inherently limited by the patient's existing immune cell fitness, really represents a biological ceiling that in vivo editing or allogeneic approaches with their capacity for optimized and consistent engineering will ultimately transcend? So in other words, is the cost problem one of autologous simply a symptom of a deeper inherent biological limitation?
Thanks, Sven. I think that's a central question, which is, when we test different donors, there's a very different efficacy in preclinical models of how well their cells work when manufactured into a CAR cell. And then if you take the variability of someone has, many lines of chemotherapy, they bring into their genetic liabilities. So I think, first of all, we've learned, this cost of $1 million per patient, that's when we have complex logistics of shipping to central manufacturing, 22-day vein-to-vein times, and so on. I think when we have things done at point of contact with a patient, logistics much simpler. We now have done trials with 3-day manufacturing with autologous cells. And in China, many CarsGen and others do fast cars doing 1 day. So the manufacturing time and the cost of autologous ex vivo cells will go down, but it's still not the logistics of an in vivo off-the-shelf delivery for in vivo manufacturing. But where I think there's always going to be a need for ex vivo is solid tumors, where highly engineered cells, at least in preclinical models, are better than single modifications. We can now, with base editors, modify 20 genes at one time in a T cell. I mean, it's amazing. So that gives you an opportunity both to make cells that are, refractory or resistant to exhaustion, to improve their function and make them traffic better and so on. I think that's a long way away from when we could do that in vivo. Now, for autoimmune disease, I think we have really very exciting data now where a single genetic modification is sufficient to have very potent effects, and also in blood cancers. That's what CAR T cells are now. So I think we're going to learn, as Jonathan said, we're going to learn the new biology with autologous cells because I think they're an established platform. But at the same time, the solid tumors, I think we haven't had any success anywhere at this point. It's pretty disappointing with cell therapies. And where I think there will be a sweet spot is highly engineered cells, which right now that I think is in the purview of ex vivo.
Right, fantastic. Yeah, really, really exciting. Thank you for that. So let's move over to Miguel. Now, thinking about this and assuming in vivo delivery or think about superior allogeneic platforms, and if they're able to achieve market dominance by, let's say, 2035, how does an entrepreneur successfully monetize the remaining economic value of billions of dollars already invested in ex vivo GMP manufacturing facilities? Does this infrastructure become specialized, highly regulated CDMO for ultra-rare diseases? Or must we anticipate significant asset write-downs and a fundamental pivot towards viral vector or LNP production for the succession of in vivo therapies?
Yeah. Thanks again, Sven. And I think we need to take one step back. And I think it's important to structure our thinking. And we've just mentioned about reclassifying what cell and gene therapy products are. And I would claim that there are three kinds of products, really. One is the in vivo, where really your product is the delivery and the payload. Another one is the allogeneic, where it's the cell, and then it's multi-engineered, as Carl mentioned. And then it's the autologous, where again, it is the cells that we deliver back to the patient with more or less engineering. And in the end, we want the same biology, which is effective cells, targeted, engineered, or optimized cells that will deliver a therapeutic biological effect. But from the manufacturing, from the industrialization perspective, it's important to have these three ideas because it's not only what Cécile mentioned in terms of the controls, but it's also where you make the investment, supply chain, the structure to deliver those products, which obviously it's kind of seems fairly easy on the in vivo. That's an advantage to deliver that biological effect. It's complex on the allogeneic, the sourcing of the cells, but we'll get there and we'll engineer those cells either to make them tolerated or to make them optimized on the biological therapeutic effect. Those will be products. I would claim that the autologous, there is a blend between the product and the process. And we may want to think about the process more as a product itself and focus on how to optimize the process. And Carl mentioned in terms of reducing the time, bringing it closer to the patient. Those are the investments that will enable us to continue to make sense economically of the biological effect that we have optimized in the autologous. And the three of them will really say, where are we going with cell and gene therapy? I've actually joked, there's a Chinese science fiction book about the three-body problem, which is we have three bodies, but we don't know where they're going to be relatively in the future, even though we know the trajectory and the characteristics of where they are today. So I would say that the process approach for autologous, the easy supply chain targeted in vivo, and the complex still to the allogeneic approach are three bodies that we do not know the trajectory, but they will three be there, and the three will bring different kinds of biological value to patients being treated.
Fascinating, fascinating. Thank you, Miguel. So that's sort of the Heisenberg uncertainty principle of cell and gene therapies. Brilliant. So, Cécile, let's come to you now. So the remarkable clinical data that has been generated by thousands of patients treated with autologous CAR T really represents an unparalleled wealth of knowledge regarding CAR T or T cell persistence, memory function, and resistance mechanisms within the human body. So as the field now pivots towards in vivo gene editing and allogeneic standardization even, what novel biomarker strategies and in silico modeling approaches do you feel are going to be critical to bridge this gap?
Good question. I still wait for in vivo data in patients to make a very, very clever answer. But I think moving to in vivo is not starting from scratch. You will use all the huge amount of data that you've been generated with ex vivo CAR T or even allogenic. So it's not something starting from nowhere, the in vivo. For me, the ex vivo data are the foundations that you will need to go faster to develop your in vivo. It's not something coming out of the blue. So telling you which marker will be the best, I have to say, I don't know right now. I still wait to see some data and see how in vivo CAR T behave at the beginning, just after infusion, but also maybe later. But I think we really should base our reflection and our clinical data and everything that we want to do with in vivo therapies on the ex vivo therapy, just to understand what is similar and not similar and to see how we can progress. But it's really the foundation of what will become the in vivo pivot, as you mentioned, for me.
Fantastic. Thank you so much. That's really, really insightful. So JP, let's come to you now. The in vivo business model fundamentally relies almost on the obsolescence of ex vivo and its manufacturing paradigm. So if we simplify the argument, the true cost driver is really the viral vector. And we discussed this in one of our previous AT3 sessions. So can an in vivo strategy realistically deliver a total cost of goods sold per patient that is low enough, so let's say under $50,000, to fundamentally displace the existing CAR T market structure of ex vivo products, or will the in vivo platform simply shift the cost burden from cell handling and logistics to high quality vector and LMP synthesis, still maintaining the high price point, but now for different reasons?
All right. I'm extremely biased, so I'm a very, very strong believer on the in vivo. And I think the in vivo really comes from the realization that ex vivo CAR T, if I'm focusing on CAR T, that the ex vivo CAR T have a huge problem of industrialization. So the scalability, that's the main problem. So we're really starting in vivo, is starting on the same premises from a biology perspective than the ex vivo. And then you're building on that to say, how do I simplify the patient journey, the hospital journey? So that's the main element. And then from the industry perspective, that's what you do. So what I can speak about, it is the experience that we have. In our case, we use a lentiviral vector approach. And in our case, we have clinical data that has been published in Nature, and then we have another paper coming in the next few weeks. The cost of goods for the in vivo CAR, so we use a BCMA CAR in multiple myeloma patients. We treat many patients. So the cost of goods is about 11,000. That's full cost of goods. So with the overhead, so we manufacture with a CDMO in Europe. So that's the cost of goods in Europe. CDMO in Europe, we ship the product to China because that's where we run the clinical study. But the total cost of goods was 11,000. And we produce our lentiviral vector in a 200 square meter bioreactor. And the next size is 600 meter. So if you are moved just from the 200 to 600, you cut the price by half. So what I'm saying here is that based on the data that we've seen, at the dose where we treated the patient and we've seen a complete response in all the patients, the CAR T is at 11,000. If I scale that up to 600 square meter, the production, my CAR T will be at 5,000 without optimizing the cell line, etc. So from my perspective, I think the goal of anyone working on CAR T, in vivo CAR T, I think the cost of goods below 2,000 should be the target, and it should be feasible, because the whole point of in vivo is to scale up, it's to industrialize the process. And what we've seen is that with our system, at least, that the in vivo CAR behaves exactly the same way that the ex vivo, the PK, etc. CRS exactly the same way. So I think in vivo is really capable, and that's a design of the platform that we need to work on, is capable of recapitulating basically the biology that you have there. But now you're solving the issues around the logistics, the issue around the lymphodepletion, which is a problem, by the way. A number of patients do not get ex vivo CAR because of lymphodepletion. We have data showing that you don't need lymphodepletion. So you're really simplifying the process. You're simplifying the whole story, and then now you can scale that up. And I'm going to push the envelope a bit further. I think where this is super exciting from my perspective is that if you are able to design a platform that is that can basically reprogram different types of immune cells, now you're getting into a situation where, for example, that was referred to by Carl, for solid tumor, CAR-T haven't been really successful. Now you can imagine a system where you have a combination of in vivo engineering, where you're engineering T cells, DC, NK, monocyte, macrophage, whatever. So you need to work on that. I know from a regulatory perspective, that's another story. But conceptually, and we had the initial data, now you are capable of really going after multi-engineering and a combination of in vivo therapy, which I don't believe you'll be able to do that ex vivo. There's no way you can do that ex vivo. There's no way. Now, if you reduce your cost of goods low enough, 2,000, 1,000, then you can have combination of different modalities, different vector or whatever, where you can treat a patient with that approach. So what I'm saying here is that I think the area of a CAR-T below 50K, we are there. So I think that barrier is going to be broken, and then we're going to have commercial products which should be produced at a much lower cost than what is currently available.
Fascinating. That's wonderful, JP. Thank you very much, and great to see your enthusiasm for the space and for what can be done. And I think, yeah, as you say, we may get into a point where the science is running a little ahead of the regulations. So let's now take a bit of a shift to Jonathan. So as we move to thinking about more in vivo therapies, and as this moves then from technical risks related to cell logistics, delivery vehicle engineering, such as viral vectors or LNPs, when you value an early stage in vivo company, how do you adjust your investment thesis to account for the dual risk of success, therapeutic payload efficiency and platform tropism and safety?
Yeah, that's a good question. In the early stage, it can be tough to talk about and evaluate value. Maybe I'll kind of take a different route in answering the question and just say that, one would try to avoid falling into the trap of momentum investing, where, investors and then accordingly companies start moving and following a theme rather than the biology, right? We saw this, following the successes of autologous CAR-T initially moving into allogeneic, because the dream of standardization, industrialization, going into iPSC-derived therapies, and the biology didn't really support that. And those, may represent a future. It's just that future is a bit further off than we thought. I think one thing that you can't deny right now is that autologous has demonstrated biology, demonstrated efficacy. Just look a couple months ago at, conference, ESMO, right? I think there was some data that was shown out of China of a bispecific CD19, CD22 CAR in B-cell lymphoma, relapsed and refractory B-cell lymphoma, 100% overall response rate, 70% complete response. And I thought that was interesting. You can't stack that risk of going after, using that bispecific, going after dual targets on top of now a unproven delivery vector and unproven payload engineering, right? I think that makes it a bit insurmountable at this point. So, when looking at and evaluating early stage companies in vivo, you're really evaluating it from that perspective of, are they going to have a truly paradigm shifting technology that can address those bottlenecks and will be able to make it to the finish line, so to speak, clinically, um, and avoiding the the allure of, okay, it's an in vivo play, so we should put our money in that. The benchmark is going to be autologous for quite a while, and the in vivo approaches are going to have to surpass that, or at least match it to a certain degree. So that's the way that we're evaluating these early stage opportunities. I think just to conclude, like when talking about the momentum investing, you could get some early exits, based on kind of the potential of a platform. If you're a new player pivoting right now into the in vivo space, I think you're late to the game, so to speak, and you're going to have to, if you're looking for an exit in this space, you're really going to have to rely on proving out the biology, extensively to the level of autologous.
Fascinating. Really, really cool. Thank you very much. Great insights. And for those of you in the audience that have got early stage companies, take note. Right, so now we're going to move into the last topic of the day. And for this one, I'm really going to toss the cat amongst the proverbial pigeons. So what if the real future isn't autologous versus in vivo, but rather autologous ex vivo combined with an in vivo enhancement or any other combination thereof, using ex vivo to create superior cells and then in vivo editing to maintain or enhance them. But combination therapies are regulatory nightmares, as JP's already alluded to, and they are somewhat funding black holes, as Jonathan's already alluded to. So are we locked into an either-or debate by our financing and regulatory structures rather than by scientific logic? So Miguel, I'm going to start with you on this one. For a dual platform, ex vivo, in vivo, whichever combination you like, is the combined COGS and sticker price inherently so high that it guarantees exclusion from broad first-line cancer treatments, locking it solely into smaller, late-stage, or even compassionate use niches? What is the maximum price or value that you think multiple products on the market can bear globally?
Yeah. So, I mean, we've already seen, and we all agree on the huge value any of these approaches giving to patients. We're discussing how to optimize the cost. And Jean-Pierre mentioned a series of elements that in vivo bring to that optimization of the cost. Carl also mentioned in terms of close to the patient, fast production. Really, I mean, those drivers will still be there and bring it down. Now, underneath all of that is the fact that I do believe that combination of some sort is going to be needed, helpful, improving the delivery of the efficacy that we want. I mean, biology, and it's about redundancy. We have multiple parallels. Medicine is about combination. And we know that about all our experience that most of the situations you will need more than one therapeutic approach to get the desired effect. So I think we're going to see combinations. And what's going to drive those combinations is that optimization of the target biological effect. What's going to hinder the combination is going to be the cost and definitely the regulatory discussions. But we're going to see it. And we're going to see it potentially in terms of autologous and in vivo. I think probably, and Jean-Pierre mentioned it, we're going to see multiple in vivo. That probably is easier to do with in vivo approach, but you may want to comment down the road. My claim is we need to start thinking about combinations for sure. Time will come for them. And as they come, yes, the value will significantly increase. The 1 + 1 in terms of combination should not be 1 + 1 in terms of cost, because we need to drive it down. It will become higher cost, at least in the beginning for sure, but will be coupled with much significantly higher value. So I mean, again, the science is fantastic. We need to make it industrially viable. And then in the end, we'll be able to optimize the therapeutic effect to bring to the patients.
Fantastic, Miguel. Thank you so much. Really, really insightful there. So, Jonathan, let's come to you, and maybe this is almost a bit of a follow-on from your last question. So, do you really, as you think about the investment side of it, do you view an integrated two components, so ex vivo, in vivo, or any other mixture thereof, system as a binary funding black hole, so twice the technical risk for a single clinical payoff? Or do you feel that the platform's ability to cross-validate two technologies inherently makes it a more robust investment? So it comes down to platform or product. Which element do you fund first and why?
Yeah, excellent, excellent question. And I guess I can keep it brief. I think, in the case here, trying to underwrite, two levels of risk, two risk curves in a sense, can be very challenging from a financing perspective. I actually think that testing the combinations is one of the biggest challenges that will be faced with cell and gene therapy. These therapies are by their nature durable and long-lived. They don't have a short half-life. So it can be difficult to test for causality between, 1 + 1 = 3. I think that, all combined together makes it very challenging in this environment to be underwriting that.
Excellent. Thank you very much. So we need to really be thinking about how do we prove that better. So let's think about the science side of it, particularly Cécile. Scientifically, does the necessary trial design for a combination approach, which must isolate the independent contribution and the synergistic toxicity of two separate interventions, does this render the trial protocols so complex that they're functionally unexecutable by most organizations? And what is the single most critical endpoint you would demand to give yourself comfort with a dual product like this?
So thank you. Well, nothing is infeasible. So you can start by saying if you want to do it or evaluate it, you will find a way to do it. But for me, you have two different way of making a combination. Either you want to retreat patients who have failed on ex vivo marketed CAR T, and I think it has already been done by people on the panel, and it's not really a combination because it's something that you use after, but it's simplifying the regulatory and the way you design your trial, because you have either a maintenance or a rescue. That can be a good way of evaluating also your product for a future combination. But if you think about doing a real combo with something that will go together, like a prime and a boost, or it's getting more complicated, because at least right now, because you don't know all the good things and less feature for the different technologies. So either you decide to put twice your own product, and maybe you will have some problem with immunogenicity or not, and you can propose that as a second injection, like a boost, or you want to combine technologies, and now you need to know more right now about the technology available. But if you have to choose an endpoint, apart from safety, you need to have a measurable, durable, functional benefit with the second injection of what you want to do right now.
Fantastic. Thank you so much. So I think that definitely sounds like another topic for a future AT3 session. So JP, let's come to you. If we think about this combination approach, and if we think that it does show this clinical superiority, would existing in vivo companies commit the necessary IP and licensing and infrastructure investments to integrate the older ex vivo component? Or is it the strategic goal purely to render ex vivo obsolete regardless of the combination's theoretical peak efficacy? So in other words, is a combination a scientific necessity or temporary competitive compromise?
That's a very good question. Look, I think the objectives that we have, it is basically to treat patients. So at the end of the day, what makes sense needs to be done. That means that the biology needs to win. The biology needs to drive the combination, like Miguel said. So it's the first thing should be the biology. That being said, if I focus just on CAR T, for example, I will have a hard time to see how an ex vivo CAR T can be combined with an in vivo one. And I don't see that happening easily. I think the in vivo CAR Ts will prove to be very much equivalent to the ex vivo. That's number one. And number two, like I said, I think today with the in vivo CAR T, for example, I can imagine treating a patient with multiple CAR Ts at the same time, not as a dual CAR, but generating a B7H3, etc. And especially if you think about solid tumor. I'm focusing on solid tumor because we know CAR T, for example, are useful to basically today only on inpatients, and that's less than 10% of the cancer patient. So we need to think about the 90% onto the solid tumor. And I think that's where the in vivo has a huge potential. I know we're looking into autoimmune as well. We should be doing that. But I think at the end of the day, we should come back really to the solid tumor. And that's where the combination will make sense. So do I see a combination of ex vivo and in vivo? Potentially not CAR T. Oncolytic virus, for example, combined with an in vivo CAR, yes, that will work. But again, there the question of the timing, the cost, the logistics, I mean, all of that needs to be considered. But at the end of the day, the biology needs to make sense, and then we can figure out the regulatory and the economic perspective.
Fantastic. Okay, no, that makes absolute sense, and I love the solid tumor, and that sort of takes us to Carl. So, Carl, the final word goes to you. If the scientific logic dictates that ex vivo priming, say, and followed by in vivo maintenance yields the most durable outcomes, do you think the current regulatory structure, essentially treating the patient's body as a manufacturing site it doesn't understand, acts thereby to block the superior science? What are your thoughts?
So I think if you look at the thousands of papers coming out preclinically, that combinations of different platforms, as you alluded to earlier, Sven, show improved efficacy. I mean, it can be an ex vivo cell therapy with an in vivo recombinant protein, a BiTE, ADC. It could also be oncolytic viruses that JP mentioned. I think we have a pilot trial we've done with an oncolytic virus given 2 weeks before a CAR T cell, and in 7 patients, it looks like it improves the efficacy in pancreatic cancer. But the problem is that we have basically, I think, a regulatory nightmare to get these things in, and it's both from our health authorities and then getting different companies to play with each other to, test and develop at risk. So I think one way to would be here to have an IIT approach like this, which China is using very effectively as a way and basically the sandbox where these kind of combination approaches could be tested. And then that would give hopefully the confidence for the investments to happen then in more formal trials. But so I think there's a huge potential there. But right now, unfortunately, it's basically a big pile up with very few clinical approaches, and it's all preclinical, which is a disappointment to me scientifically.
Yeah. No, that's really insightful. Thank you very much, Carl, and completely see where that challenge comes. And I think very often we used to talk about with all these different therapies, it's about a toolbox, but yet the regulatory system appears to be pushing us towards I have a hammer, so everything looks like a nail approach, and so it's a once only. And I think the other side of it is the pre-competitive space, that ability to work in that, and that our last AT3 session actually explored some of that. So I think that's a phenomenal example. Right. So clearly we're living through an evolution of a cellular revolution. The science is moving so fast, whilst clinical development and commercialization activities and regulatory, as we've heard from Carl, really struggle to catch up. As we're here not for ourselves, but for the patients we serve, it really behooves us to continually challenge and test what we're doing, where the science is going, and so ensure that we always keep our eye on the patient and encourage regulators, manufacturers, and everyone else to do the same. So now that we've got a few minutes left, I'm going to hand over to Lee, who has promised me some really cool questions from the audience. So Lee, would you like to lead us off on questions?
Yeah, outstanding. So there's been some super interesting chat and also some questions coming in. I'm going to do my best to lump some of these together, and we're going to do this really sort of rapid popcorn style if the panelists are willing. So one of the themes I see emerging in the questions is, we talk a lot in the ex vivo autologous cell therapy setting about patient variability, cell variability of the starting material. That's obviously no different in the in vivo setting. We have patients with exhausted T cells in vivo, and in vivo is going to come across those same challenges. And then on the other side, we see CAR-related toxicities for ex vivo autologous cell therapies. Do we imagine there's going to be the same kind of, profile of CAR-related toxicities in vivo? How do we manage the patient variability on the in vivo side that we've seen on the autologous ex vivo side? I'm just going to throw it open.
Who wants to dive in?
What? I think indeed for ex vivo, one of the differences between ex vivo manufacturing and in vivo for CAR-T, for example, is that I think for the ex vivo, you have an opportunity here to take the cells out of the patients, and then you culture them, etc. You can even tweak a little bit the CD4, CD8 ratio, and so on. You can play a little bit on that, which you cannot really do in the in vivo unless you start to engineer a bit more. But one thing which is important is that when you're doing in vivo, you are using resident T cells. You don't exhaust them the same way than you will do in ex vivo culture. With ex vivo, you need to culture them a few times, etc. Most of the times, right? So I think the in vivo cells are a bit more fresh, a bit more, but it's true. Those patients have seen many treatments and so on. So you cannot really control that. From the clinical trial that we've seen, like I said earlier, the PK of the in vivo CAR and ex vivo CAR are exactly similar. So from a CRS perspective, you see exactly the same type of behavior. Around day 10, 12, you have a CRS grade 1, 2 in our case. So it's going to be the same type of toxicities that you will see. One thing that we've seen with lentiviral vector, at least, it is an immune reaction to the lentiviral vector, expected at day 1. Because you're injecting a lentiviral vector, so it is expected. So you see that reaction to the lentiviral vector injection. So that's the observation that we've seen.
Anybody else a quick chime in on that one?
Well, I think Jean-Pierre said it. I mean, we would expect the same thing because we want to do the same thing. Is really the control of the final cells, which become the product, versus what happens in vivo, because you just deliver. So you kind of do the manufacturing ex vivo or in vivo, but then in the end result and the beginning of the starting material brings to a series of similarities on the efficacy, on the outcome of the cells, and I would say also on the safety aspects as well.
So many great questions. I wish we had another hour for the questions. JP, I'm going to flip one quickly to you. Are we seeing and one of the things I'd like to say about investors is they act irrationally and then like to blame the industry for their irrationality. But are we seeing an in vivo bubble?
There's a strong excitement for in vivo. Is it a bubble? Potentially. I think now you're seeing a few things where you're just like, no, I don't think that's going to work. But I think the industry has been like that. I mean, when there's something that works, there's a lot of excitement, etc. A few years ago, when we started with the in vivo pitch, I mean, nobody believes into that. There was a lot of skepticism. Now the data are coming in and we're seeing this is working and it's going to work. So obviously, a lot of money is coming in. Yes, there's some sort of excitement, but I think over the next 5 years, we're going to see more and more clinical data. Number one, we're going to understand much better the tox profile, etc. And then things will settle down a bit more.
Jonathan, from the investor point of view?
Yeah, I take offense to your earlier comment there, but
of course you do.
Good, well done.
Yeah. No, I would say, it can feel maybe that we're generating a bubble. There are multiple factors in play, right, which kind of direct investors and where they're going to go. It's, we have our own investors as well, right, that we're trying to create narratives for and generate returns for. Um, I would say one thing that does feel a little bit different now is that we are seeing some promising biology, right? That's being generated and some promising data that's being generated. And I think that's going to ultimately be the difference maker.
Thank you. I sadly have to turn it back over to you, Sven.
Thanks, Lee. And Lee will also put together all of the thoughts and summaries, and we will make sure that we send this out with action points of where we can go and how we can work on this. I'm going to speak fast because we will get cut off at the top of the hour from previous experience. So I want to thank all of our panelists and all of the audience for all of your engagement. Thank you so much. Also, a huge thanks to our season sponsor, BCA Advanced Therapies Network, and our session sponsor, that is Scale Ready, for supporting this work. We're completely unpaid for this, and that money goes towards paying for the sessions online and things like that. So thank you. Also to Beacon for sharing the exclusive data and insights at the beginning of this session. So as you may be aware, this session is just one of a part of a much larger conversation, and we really need each one of you to help carry it forward. Cell and gene therapies hold so much promise of transformation for patients, but without affordability, that promise remains out of reach. If you'd like to register for future sessions, contribute to ideas, subscribe to our mailing list, please visit us on advancedtherapiesthinktank.com or follow us on LinkedIn. So until next time, challenge convention, build transparency. Let's get these therapies to patients that they really, really need. And remember, we will not have a session in January because of JPM, but we'll be back with a post-JPM session, I think it's the 5th of February. So we haven't been cut off at the top of the hour, which I'm really surprised about. So Georgie must have done some magic. But on that, thank you very much, everyone, for joining us, for your contributions, for putting up with me with my crazy questions, and for keeping to time. So thank you, everyone.
Thank you, Sven.
Thank you,
Thank you.
everyone.
Thanks, everyone.
Yeah, thank you,
Lee.
panelists.
Bye-bye.
Thank you, everyone, for
Lots
great
of fun.
questions.
Bye, everybody. Thank you.
Bye.
Bye.